Alright, this is the continuation of mini lecture 20.1b because we're having some technical difficulties, but then you have nice really mini lectures. So, again, looking at the thermodynamics of nucleophilic addition elimination, here is an example. If we want to try making an anhydride, in this case this is called a mixed anhydride, by adding this carboxylate to our amide up to the tetrahedral intermediate, the electrons swing back down and kick out N-, we can certainly compare the stability of our negatively charged amide here to that of the carboxylate. We know this is really much weaker and much more stable, so we know this reaction does not proceed as written. You can't make an anhydride from an amide. Amide is at the bottom and extremely stable at the bottom of the reactivity ladder. Well, turns out that's kind of a good thing, because as it happens, we are amides. We are full of amides. We're full of proteins and peptides. In fact, proteins are polyamides, as you can see here. I'm circling all the amide linkages. And if those really were very reactive, well, we would not be around to tell you about it. So amide stability is actually a good thing. Another really cool thing about reactivity, the reactivity ladder, and this kind of chemistry is barely covered in your textbook, but it's worth looking at because it's so interesting about how Mother Nature can get around these issues with thermodynamics. So it turns out we need glutamine. It's an essential amino acid, and it's synthesized from glutamate. But if you look at this biosynthesis, it shouldn't work, because what we're trying to do is substitute a carboxylate to make an amide, and this is definitely more stable. This reaction is not going to happen. So how does Mom Nature do it? Mom Nature does it by adding a great leaving group in the form of ATP. So ATP, this is biochemistry's form of a leaving group. These are basically biochemistry's version of a Cl- or a tosylate. It's just what Mom Nature uses. So what Mom Nature is doing, what happens in this biochemical pathway, is that glutamate gets phosphorylated to, voila, put a fantastic leaving group on the glutamate. In that case, then, the nucleophile can add. We get to our tetrahedral intermediate, just as we do in a beaker, but now, yay, the tetrahedral intermediate can collapse to kick out phosphate, a fantastic leaving group, and make glutamine. So there's lots of interesting applications of this kind of chemistry in biochem. Here's another interesting application of this chemistry, and it's obviously biochem because that's obviously part of what was once a living organism. That's a big old hunk of fat, and back in the day, our pioneer forefathers and mothers, most likely mothers, would boil that up in a big old vat of lye, aka hydroxide, to make soap. So basically this is an ester in the form of fat, and it gets hydrolyzed under basic conditions to give you soap. How does this happen? Well, this is fat right here. This is what makes up fat. This is a TAG, or triacylglyceride. And what you can basically see is that there are 3 ester linkages in there. And if I treat this with my hydroxide, I'll do the old up, down, out thing 3 times, and I will get my alcohol, which in this case happens to be glycerol, hence the name triacylglyceride, and my 3 molecules of my fatty acid. So esters are always made up from an acid and an alcohol. It's just in triacylglycerides there's only 1 alcohol that adds 3 acids. Well, these fatty acid that are saponified are under basic conditions, so they're negatively charged there. And that means, nifty grifty, I have a very polar, ionic, hydrophobic piece to my carboxylate here, but I also have a long, greasy, hydrophilic tail. So greasy tail plus polar head equals soap. And that is because dirt and oil and that crud on your plates and in your clothes is something that's more non-polar. So let's make this red or orange here. These are the greasy tails that happily surround the dirt and oil, but exposed on the outside are your polar heads, and so they solvate this whole thing in water. These are micelles, and basically this is all review from way back in OChem 1. It's pretty important though, and you'll see it again. If you've seen it for sure in cell bio. If you took cell bio, you'll see it again in biochemistry. These long greasy tails and polar heads are again a pretty predominant part of our lives, in that phospholipids have long greasy tails, as evidenced by these little squiggly blue lines. And now, instead of a polar carboxylate head, we have a polar phosphate head. And those phospholipid bilayers are sort of important in that they basically make our cell membranes and keep us alive. So, cool stuff. But back to saponification. It's an irreversible reaction. Why is it irreversible? Well, it's quite evident, and again, great review. If we start looking at the reaction coordinate energy diagram, we know that our hydroxide here will add to give us our tetrahedral intermediate. This is not exactly the same molecule, but you'll get the big picture here. This tetrahedral intermediate, a carbon with 3 oxygens on it, that is highly unstable. So it would be very facile for the reverse reaction to take place. So this first step is absolutely reversible. But, of course, the tetrahedral intermediate can choose, if you will, to collapse to make the carboxylic acid, kicking out the alkoxide. So here's my alkoxide. And of course, I can stop at this point and say, all right, fine and dandy, but if I look at this, it took me this much energy to get up to this intermediate, and there's clearly that much energy here, so I would expect this to be reversible. But wait! As you can see, there is another option here with an extremely small delta G double dagger. So it's going to be exceptionally fast for my alkoxide to deprotonate the carboxylic acid that was formed to make the very stable carboxylate. And so, in essence, this is irreversible because we'd have to go all the way back up to here. We're at a very low level thermodynamic sink, very stable. Saponification is absolutely irreversible. Alright, a couple more examples. Esters can be hydrolyzed. under acidic or basic conditions. So we'll look at an example under acidic conditions to wrap this up. Of course, the first thing we would expect to have happen would be protonation. Where? Well, you know, here, there, and everywhere. We're going to put it here first, see if that gets us anywhere that we want to go. I'm going to just call this OPR for propyl. Now, it's instructive to look and see where we're trying to go. We have a propyl ester here, and this isn't actually a hydrolysis if I use methanol. So I'm going to use water, because if I want to hydrolyze an ester, I am changing the ester back to a carboxylic acid. So my nucleophile is going to be water. And so, of course, you would expect it to add here and make our, I'm just going to call this AR to not take up so much room. That gives us our tetrahedral intermediate. And if I'm trying to hydrolyze it, I have this huge excess of water. That means, I guess I am ultimately going to be losing the loser group here, because it is going to be replaced by this large excess of water. Well, if I'm going to replace it, then I guess that means at some point it had better be converted to a good leaving group by protonation. And as we are wont to do, if we see something that's protonated, we're going to deprotonate it. So now I can cheat because I have this nifty eraser. I'm going to deprotonate here, and I'm going to show this protonated intermediate. And now what? Well, now it's quite evident, I hope, that I can just take my water and flip that down and kick that out to get my hydrolyzed ester. And so the reason we can do this is because this guy must be present in huge excess. And even then, we're going to see that there's going to be some, there's no way we're going to get 100% yield on this. There will be an equilibrium. So that's an example of acid-catalyzed hydrolysis of esters. Here's an example with a really complicated ester. This is erythromycin. If you look slowly and carefully, you will see there is my ester. And so, in the body, when I ingest erythromycin, one of the reactions that's going to take place is protonation at the ester, addition of water, and I will hydrolyze my erythromycin to make, of course I'm not going to draw the whole thing. All this comes over here. This acid, well, let's see, I'm going to have to back it up here. There we go. And that's just one of many steps that's going to take place in the metabolism of this particular drug. So, that's plenty on hydrolysis of esters. Next up, you will see, we're going to look at some more specifics, that being looking at acids, amides, and the Gabriel synthesis.